CoA, so you have acetyl-CoA. So right now we only talk about one of the two pyruvic acid. Okay, so this is one turn only. Okay, so then I'm gonna go back to, well, I'm gonna keep it here. So the two carbon molecules, I don't know why it needed to be added to a 4 carbon molecule called oxaloacetate. Okay, so to make citric acid. And now the enzyme, each of this is controlled by an enzyme. There's a different enzyme in here. Okay, so then the enzyme can bind this and some stuff happening. Okay, so one of these, let's see, 1, 2, 3, 4, 5. So you notice one of these bond is broken. And again, when it's broken, you get some NADH out, and then one of those carbon becomes CO2. Okay, and then they're gonna break it again. So now 1, 2, 3, 4. Okay, you get more. Okay, and then this step is just rearrangement. Okay, removing a acetyl-CoA, which you don't need to know. Okay, you make a GTP. A GTP is equivalent to an ATP, so it's a 1-to-1 ratio. And then more rearrangement happen. Okay, you get another molecule called the FADH2. Okay, and then just rearrangement, and you just get more high energy electron, and they give you back these molecules. So, but what I'm asking is this. Okay, so what's coming in? What's coming in will be acetyl-CoA. What's coming out will be 3 NADH, okay, 1 FADH2, 2 CO2, and 1 GTP or ATP. This is per pyruvic acid. So this is gonna do 2 times because you have 2 of these. Okay, so that's why for this you need to double. So in the end, for per glucose molecules in the citric acid cycle, you get 6 NADH, 2 FADH2, 4 CO2, and 2 ATP. So that's the Krebs cycle.
Okay, I got it. And from my understanding, it starts off like glycolysis right first, but if it has oxygen, it'll go to the Krebs cycle, and if it doesn't have oxygen, it'll go to the fermentation cycle?
No, Raymond, remember we also have the anaerobic cellular respiration. For anaerobic cellular respiration, they do still go through the citric acid cycle and the electron transport chain.
Okay.
Yeah. But if an organism do not have the enzyme, because remember these are all enzyme regulated. So if a bacteria do not have the enzyme for the citric acid cycle, and if there's no oxygens, then the bacteria will do fermentation.
Okay, so it's determined by the enzyme of the bacteria if they go through the Krebs cycle or not?
Yes.
Okay.
Yeah, so in general bio, we're trying to make it simpler by just saying if they don't have oxygen, they do fermentations. But really, it depends on the enzyme in the citric acid cycle, whether they're there or not.
Professor, in Krebs cycle, what's in?
I'm sorry?
In the Krebs cycle, what's coming in?
Acetyl-CoA.
And both acetylcholine coming in?
Correct, because if you go back, you start with
What's
6
the difference? There is nothing different?
Between what and what?
Between Krebs cycle and this cycle.
Like the Krebs cycle is the citric acid cycle. They just name them differently. Krebs named after the scientist's name, and then citric acid is named after that molecule. The cycle is the same. It's the same exact thing. They just name them differently.
Okay, thank you.
Mm-hmm. Alondra.
Okay, and then going, adding to that, because I know on the study guide, it says what is the role of NADH, NAD plus? What is their significant? Why is the importance of pre-oxidized NADH back to NAD plus, FADH2 back to FAD plus?
Mhm.
Could you go over that?
Anyone want to give that a try? What are these?
Are those called electron carriers?
Yeah, the high energy,
Yeah.
right? Because each of NADH in the electron transport chain, it changed to 3 ATP. And each of this is worth 2 ATP. So to our advantage, it is better to make these guys than just ATP. Because these have more value.
Question, Professor, how would you, if you're okay with sharing this, how would you word that? Because some of the questions on the study guide, as I was trying to answer them, I got a little confused. And then when I was going and doing further investigation, it was very simplified. Like, oh, okay, that's how it is. This one stumped me, and I still have yet to answer it. How would you word this particular question for Chapter 5 study guide for question 4?
Is that the one that Alondra asked? Is it the same one?
Yes. Because I know you were describing it, but how would you word something like this?
Okay, so the first part of that question is, what are they? And so we are saying, and Raymond say, these are high electron energy carrier molecules. They have more energy value than ATP. The second part of that question was, why do we need to go backward? Why do we need to oxidize them to get FAD and NAD plus?
Because they need to be recycled, right?
Yeah, why do we need to recycle them? If you don't have these,
So
will this work?
continuing. So
Right.
because if it's stopped, there will not be any, another reaction. So it has to be continuous.
Yes, perfect, right?
But
Not
I
just
have a question for that. So for me, NADH and FADH2, are they enzymes or electron electron carriers? Like for me, what are they?
coenzyme.
Coenzyme?
What is the function of a coenzyme?
The
It's the
function
non-protein part of a enzyme, right?
Yeah, you guys,
No, it's a cushion for the enzyme to be placed, I think.
right here.
Yeah.
This
Not-
guy.
Assist.
Enzyme is a catalyst reaction.
They not only make the active site fit the substrate to speed up the chemical reaction, but they also involve. When you break bond, you release electron and hydrogen. Who's gonna pick those up? Because if they are not being picked up, electrons are very reactive. It's gonna cause damage to the cell. Hydrogen, if not left pick up, is gonna change the pH of the fluid in the cell. And you guys know, a change in pH will mess with the enzyme activity. So we are lucky we have these guys. NAD8, NAD plus, FAD, NADP, and coenzyme. All of these act as coenzyme. They not only make the substrate fit better, that's why we put coenzyme A, acetyl-CoA. Okay, so that they fit the active site better, but they also help carry away the electron and hydrogen. That's why we call them high energy electron carrier. Those electrons have very high energy. And so we make them, and then in the electron transport chain, that's where we dump them and recycle them back. Okay, so why? Look at this, glycolysis need NAD plus to make NADH. citric acid cycle need NAD plus FAD to make these. So you need to recycle them. If you don't have a substrate to keep these going. This whole thing gonna shut down. We're not making ATP. Okay, if it's us, we're in trouble. If the bacteria gonna be in trouble as well, but not as much because they able to do other stuff to survive. We are more limited. So I think that's the answer to that question. Did I miss anything? It's like, what are these?
I was a little confused, but I knew that it is coenzyme, but I was not sure 100%. But can you go over, please, the pentose phosphate pathway?
Wait. I think Raymond asked the electron transport chain as well.
Yes.
And then we'll come back.
Okay, thank you.
Okay. But then I'm trying to answer the question in the study guide, the one that Aaron and Alondra asked. I'm trying to answer that. Okay. If I remember, the first part is what are they? Okay. And then the second part is why are we going backward? Okay. We need these to keep everything going. If we don't have it, everything will shut down. I think that's all right. Alondra, is there anything else on that question?
I'm sorry, could you repeat? You said they are high electron energy carrier molecules.
Yeah.
They have more, you said energy value?
Energy value, yeah.
Okay.
Mm-hmm.
Thank you.
And then the second part is, I think, why do we go backward? Why we oxidize them back to Na?
They need to be recycled so that way they can continue the cycle, right?
Right. Not just this, but glycolysis, the intermediate steps, and all that.
Okay.
Mm-hmm.
Thank you.
Is that complete? The question? Should I answer all part of that question?
You did, Professor. Thank you so much. By answering that, you clarified so much and opened my mind in more ways. So thank you so much, Professor.
Of course. So then now we gathering all of these, right? The cell will not use these. The cell said, you need to change it to ATP. Okay? They're like, you take all of those and you go somewhere and you change it to ATP, and then I can do work with you. And so along the way, we gather all of it. Okay. Here from glycolysis, and then all of these from the Krebs cycle. Between glycolysis and the citric acid cycle, who make more NADH and FADH2?
NADH.
Between glycolysis, right?
Mm-hmm.
And the citric acid cycle, between the two processes, which one will give the cell more in terms of number, NADH and FADH2?
Oh, the citric acid, the Krebs cycle.
Right. So that is the purpose of the citric acid cycle. I think there was one question say, what is the significance of the citric acid cycle? Okay, is to make NADH and FADH2, these high energy molecules. Then we gather all of those and we go ahead and head over to the electron transport chain. Okay, so the first protein complex will remove a hydrogen from NADH. And now look, we recycle, we oxidize NADH. We recycle these. These now can go back and keep glycolysis and the citric acid cycle going. Okay? And look, the next complex, Q. Q does this, almost the same, removing H plus an electron and recycle FAD. So I think you have the Flearside case study, right? Did I give you guys that one?
Yes, you did.
Yes, you did.
Okay. So if you've done that study, you realize that the Flearside, okay, is a chemical to kill flea on dog. The Flearside is a non-competitive inhibitor of this complex. Is a non-competitive inhibitor. which means it's going to bind at the regulatory site, and the site where NADH is supposed to bind, it cannot bind anymore. And so you have NADH, but this doesn't happen. So this is not recycled. And that's why the girl did not survive, because her electron transport chain did not do this. She did not get NAD plus. Okay? And so she can't continue glycolysis and citric acid cycle. The other detrimental result of this is she not making ATP. You need this and this to drive the electron transport chain to make 32 to 34 ATP per glucose. The problem is it stopped here. So none of this happened. So she is not making ATP. In human, we breathe and our heart pump and our brain work because of ATP. So if this is broken down in her system, she's not making ATP. That's why I think her oxygen level dropped. Okay? Because her diaphragm stopped working and she's not getting oxygen in. And the brain used 15% of the oxygen we breathe in. And because her diaphragm stopped working, her lung not getting oxygen, her brain not getting oxygen, it not sending signal to the rest of the body, and everything just shut down. So this answered the other question, why this need to happen. Okay, to recycle this, to keep everything working so that she constantly get ATP. Same in bacteria. Okay, bacteria gonna need ATP to power all the process that keeping them alive. Okay, so they have this as well on their plasma membrane. So anyway, the electron transport chain, the first complex will remove electron and hydrogen from NADH. The second complex will remove H plus and electron from FADH2. Okay, and like I say, movement of electron is power. Okay, so you have these complexes move electron. And when it move electron, it power these are pump. So power them up and they're gonna pump the hydrogen to the other side of the plasma membrane. So ultimately what they're doing is creating a concentration gradient. Okay, high, H plus out here, low in here. That is called potential energy, the ability to potentially do something or do work. Okay, the cytochrome C oxidase in lab we test oxidase, right? We do the oxidase test. That's what we're looking for is the cytochrome C. Sorry, this one. Okay, and so the cytochromes, the cytochrome A3, okay, will take the electron, very high energy, okay, and combine with oxygen and hydrogen that came back down, okay, the hydrogen floating in here, and it formed a more stable molecule that all living thing on Earth need, which is water. Okay, so that's what the this cytochrome does. Okay, change into water. For aerobic cell respiration, the final electron acceptor is oxygen. Okay, and then this last complex is called the ATP synthase. Okay, this complex is going to open up, and then when it's open, okay, hydrogen will naturally go from high concentration to low concentration, and when it does that, it actually power this up. And I don't know the mechanism. I have to be a biochemist to understand this. Okay, but in there, what it does, it uses that energy to make the bond between ADP and one of the inorganic phosphate that's floating around, make that bond, and now you have ATP. So for every one NADH, you get 3 ATP. For every one FADH2, you get 2 ATP. So we gather all of those, we funnel into here, and we don't just have, the bacteria in us don't just have one of these sitting around. throughout the membrane, you have many electron transport chain. And so very quickly, we're going to make a lot of ATP for every molecule of glucose we consume. And same with bacteria. Aaron has that. I'm sorry, Raymond has that.
Thank you so much. I understand it now.
Okay. Now, question on the electron transport chain? All right, now we're going back to this. Okay, so these are additional